Welcome to today's Help Me, I'm Confused biology review session. We're going to talk about the enzyme telomerase and how it prevents the lagging strand of DNA from getting shorter and shorter every time the DNA is copied. What we see here are the two strands of the template DNA at the top and at the bottom, which were the original DNA, and then the two new strands shown in green. The one on the top is the leading strand. It's just as long as the original template strand. And the one on the bottom here is the lagging strand. Right now, it's as long as the template strand because the RNA primer hasn't been removed yet. When the RNA primer is removed, we see that the lagging strand is shorter than the template strand. This is, of course, because we needed to remove that RNA primer. And remember that the lagging strand is synthesized in fragments called Okazaki fragments because the 3' end is at the opposite end of the DNA from the replication fork opening, which in this case would have been in this direction. So what do we do when we remove the RNA primer and the lagging strand is shorter than the template strand? This might not mean very much initially, since we discussed that the telomeres, or the ends of DNA, don't contain very many genes, but eventually, if you kept on getting shorter and shorter with your lagging strand, the older you got, the more DNA you would lose, and you would start to lose important genetic information. So instead of allowing that to happen, what we have is an enzyme called telomerase. Now, I just mentioned that the ends of DNA don't have very many genes that code for proteins. They do have repetitive sequences of DNA. Telomerase is an enzyme that carries around its own little piece of RNA primer, and that RNA primer is complementary to that repeating sequence that we find at the end of the DNA. So you'll see here that the telomerase has come in, it's able to localize to the telomere region of the DNA, and the RNA primer is hydrogen bonding to the complementary nucleotides on the template strand. What the telomerase actually does is not to extend the lagging strand, but it extends the template strand, which we could see here. And it does this by adding in nucleotides that match to its RNA primer. So essentially, this RNA that the telomerase carries around becomes the new template and is used to extend the original DNA. Now, this is important because once telomerase leaves, we now have a longer template strand on this strand of the DNA. So now we could go forward with the normal process of DNA replication, where we have primase laying down an RNA primer that is complementary to the template strand. And once the RNA primer is laid down, we have a free hydroxyl group. And DNA polymerase can come in as usual and just extend this. All right. So just as we did in the beginning, we're going to remove the RNA primer. And now we end up with the 5' end of the lagging strand there. And although the lagging strand is shorter than the template strand, it is the same length as the other two strands, the leading strand and the other template strand. So we're not losing any important genetic information here. This part of the DNA of this template strand remains single-stranded. But that's okay, because remember, this just consists of repeats and doesn't contain any DNA that codes for genes or proteins.